Hello and welcome to Chapter 10, Meiosis and Sexual Life Cycles for EFB103. Let's get started. All right, so we're getting into genetics now. This is a new unit in your textbook. This is the first chapter. And this connects to the previous chapter on mitosis, thinking about cell division, but a specific kind of cell division that leads to some of the interesting variation that we see. And you can take a look at this photograph here of this nice family. And when you look at a family, you might notice that offspring tend to look like their parents somewhat, but the kids look a little bit different. So there's variation. There are traits that are passed along, but there's also variation in those traits. And so we'll see what underlies that at a cellular level. Now, it's helpful to introduce some general terms and ideas as we get into this. Probably most of these things you're already familiar with. So heredity is the transmission of traits from one generation to the next. Variation are some of those differences that you see in offspring relative to the parents and the offspring relative to each other, so between siblings. And then genetics is the study of these two things. And so ultimately, it's genes that are being passed from one generation to the next. And so genes are this unit of heredity. And we've already talked about chromosomes to some extent, and we'll talk more about this in later chapters as well. But genes are found at certain positions on chromosomes. So you can look at this example here, chromosome number 17. It's marked with some specific genes along where they occur along its length. These are just some examples. So chromosome number 17 has over 1,000 genes on it. Too much text to show on here, but so these are just some examples of where these occur. And so that position is called the locus, or if we're talking about more than one, we say the loci. Now, another important fact here is to understand that most of your cells, those are the somatic cells. So basically, somatic cells are everything except gametes, which are sperm and eggs. You have pairs of chromosomes. So say chromosome 17 that we just looked at, each of your cells has two chromosome number 17. One was inherited originally from your father, one inherited from your mother. And so they're often called paternal and maternal. And they occur in pairs, and these pairs are called homologs. And each homolog is going to have the same genes, just like we saw in that map of chromosome 17. They occur at the same locus, but there might be differences between them. So here, this heterozygous gene here, little c, big C, that's just annotation that we often use. These variants, these differences are called alleles. So each chromosome has an allele, and sometimes those alleles might be different. Now, humans have 23 homologous pairs. We have 46 chromosomes overall. Again, but 23 pairs of them. So different from somatic cells are the gametes and the cells that directly produce gametes, or are called germline cells. And so what's happening in the case of gametes is they contain half the number of chromosomes. So they only contain one homolog. So, a gamete is only going to contain one chromosome number 17. It doesn't contain the homologous pair. And so these are sperm and eggs. Those would be gametes. And this is critical in these sexual life cycles. And we'll get to that in a second. What about asexual life cycles? First off, because this is more or less what we've seen previously, a cell here, like this amoeba over here. Here's a parent cell. The genetic material is replicated, and the cell undergoes mitosis, and now you have two. There you go. That's asexual reproduction. This could also be considered clonal reproduction. So each cell, each product daughter cell, is identical to the parent cell. So these are just clones of what you started with. Contrast this with sexual life cycles. Here we see that segregation of chromosomes into gametes. So here's an egg and here's a sperm. Each one of these, again, contains half the number of chromosomes as the somatic cells, and they only contain one homolog. And so these are called haploid. And so haploid is considered the N number. So humans' N number is 23. So a sperm or egg is going to have 23 chromosomes. A diploid cell, which are somatic cells and tissues, those are 2N, and they contain 46 chromosomes because you have the pair of homologs, not just the single homolog, but you have two chromosome number 1s. You have two chromosome number 17s, one that's paternal, one that's maternal. And so our total number of chromosomes in most of our cells is 46. Now we know how we can visualize these numbers of chromosomes. We've talked about karyotyping before. So here's an example here of a scientist. She's looking at this cell. Here are the chromosomes condensed. It's important to notice that we get these karyotypes from cells in metaphase. So at this point, all the DNA in that cell has been replicated in S phase of interphase. We've got the pairs, sister chromatids. So these are direct copies that have been made. But if we look, we also have those homologous pairs. This is where sometimes people get confused looking at, well, wait, you got these two red ones. Isn't that a pair? Those are the sister chromatids. Those are just identical copies. But these originated from the homologous pair of one red chromosome, one blue. It's just they're copied because it's in the middle of mitosis, basically. All right, so we have a couple of names for the different kinds of chromosomes that we see. Almost all of them are called autosomes, but there are, in many species, and humans included, there are chromosomes that are sex chromosomes that determine the biological sex of an individual. And we can look at the karyotype of, let's see, here we got a human genetic female karyotype. So chromosomally, this is a female. And here we see all the autosomes. So there's 1, 2, 3, 4, etc. And here's that last pair of sex determining chromosomes. These are called the X chromosomes. And so this is a genetic female because they have two X chromosomes and there's no Y chromosome. Let's contrast that with a male. Same set of autosomes. Obviously, if it's a different person, they're not going to be identical genetically, but you still have the same chromosome number. But here we have one X chromosome and one Y chromosome. So this would be a genetic male. And this is important when we think about gamete formation too, right? Because you only get one of each homolog into a gamete. So the gametes a male produces, half of them are going to contain an X chromosome, half of them are going to contain a Y chromosome. All right, so let's think about this karyotyping. So we've got an organism that has 46 chromosomes. That's all we know. You're just working away in the lab, and this is the result you come up with. What can you interpret from this regarding this organism? Okay, yeah, I mean, more or less can determine that it has 46 chromosomes, and therefore its gametes would have 23. You don't know if this is a human. It could be some other organism that has 46 chromosomes. And so based on the information, that's the one thing you can confidently conclude. So let's look at these sexual life cycles in more detail. We've been talking about gametes and that are haploid cells. So the fusion of these gametes, as we see here, is called fertilization. All right, it says union here. And the first cell that's produced as a result of that is diploid again. So we had these haploid cells, and now they come together, and now you have a diploid cell, right? Because you've now got the two sets of homologs, chromosomes, again. This first cell, has a specific name. It's called a zygote. And then now we'll start to see mitotic divisions. So just think growth and development into, say, in human, in this case, a multicellular organism. And then back around, gametes are produced by specific tissues within these individuals to produce sperm and eggs. Now, different organisms that have sexual life cycles do this really in different ways, although generally the process is the same. It's more that the time spent in being haploid or diploid is different. So here in panel A, this is what we've been talking about. These animals, vertebrates in general. Here's us. So we're diploid multicellular organisms, and we have these stages, gametes that are produced in meiosis, and these are these single-celled stages, and it's sort of a short term in that stage in terms of development. That's probably the best way to think of it. And then it takes a fusion event, fertilization, to produce a new diploid organism, starting out with that single-celled zygote. Now, other organisms, as we'll see, do this a little bit differently. So let's start with plants and some algae. And I'll say this isn't all plants. So some plants, some algae, they undergo what's called alternation of generations. Now, we still see this same pattern, but the difference here and why we call it alternation of generations is we have, these diploid multicellular organisms, just like we saw with the vertebrates, but they also can have a multicellular organism in the haploid stage. So this sporophyte stage, this is the diploid stage, produces gametes through meiosis, and some of those will undergo mitotic division. So they're just copying. Think just growth and development. So we have a multicellular organism. This is called a gametophyte. And this, makes sense, right? This is almost, think of it, it's been produced from gametes, so it's called a gametophyte. So it's multicellular organism that is haploid. These can produce gametes by mitosis. Don't need to use meiosis because these are already haploid. So just producing gametes by mitosis, they fuse, come together, fertilization, zygote, we're back to diploid. And so that's called alternation of generations because you've got these multicellular stages in both haploid and diploid. The third general kind here, there's going to be variations on this if we look at all the amazing diversity of living things on this planet. But this third type that we're talking about in this class covers most of the fungi and some protozoans. And what we see here is a very temporary diploid stage. So it's almost, you sort of think of a little bit opposite of what we saw with the vertebrates. And so in this case, you have a zygote which will almost right away undergo meiosis, producing gametes. So it's just very temporarily this diploid organism and undergoes meiosis, producing gametes. Here we see mitotic divisions to produce a larger multicellular organism, or in the case of protists, they're not necessarily multicellular, but these gametes are produced by mitosis here, because again, haploid produces haploid just by mitosis, don't need meiosis here. Fusion of gametes, fertilization event, and round we go. So it's, different amounts of time, or the preponderance of the life history of certain organisms is spent either as haploid or diploid. It depends on which kind of organism we're talking about. So let's look at this process of meiosis. You already have a sense of how this generally works. We're going from a diploid cell to a haploid cell. So the daughter cells here are not going to have homologous pairs of chromosomes. They'll just have one copy or one homolog of each chromosome. That's why they're haploid. So here we see a diploid parent cell. So this could be a germline cell. The chromosomes are duplicated, and they'll undergo a couple of rounds of cell division, first splitting the homologs and then the sister chromatids. And there's a couple of really important features that happen along the way, and we'll get into those details. And so this is the broad view first. And so let me ask you a question. Consider the amount of DNA relative to a resting cell, so a cell that's not dividing, is just sitting there doing its thing, just before mitosis or meiosis begins. So this comes back to the cell cycle and understanding that. So which statement given here is most accurate? All right, yeah, so just before mitosis or meiosis, the DNA has doubled because it's gone through S phase and interphase, and the DNA has been copied. So that's the same in meiosis as it is in mitosis. All that interphase stuff is more or less the same leading up to this cell division process. And so we can see this here, interphase occurring, and then we get into prophase, metaphase, etc. as part of meiosis. Another thing to point out here is that meiosis, as it says in the middle, is not a cycle. Now, it's part of a sexual life cycle, but to bring this back around, these gametes have to fuse and produce that diploid zygote. All right, so let's look at this a little more simply. This is a figure from your textbook. So we have in a resting cell pairs of homologous chromosomes, and those will in S phase be replicated. So now we have sister chromatids. In meiosis 1, we'll see that homologs separate. And then in meiosis 2, finally sister chromatids separate. Again, now we have these haploid cells as a consequence. The phases of mitosis look similar to what we saw with mitosis. So again, interphase is going to be pretty much the same. And then we have meiosis 1 and meiosis 2. Meiosis 1, this is where a lot of the important action happens. So that's really what to pay attention to. Meiosis 2 resembles a mitotic division in the sense that here is where we see sister chromatids separating. A final point that I'll add on this slide, I've added here just in parentheses, pro-metaphase 1, pro-metaphase 2. So pro-metaphase is something we saw with mitosis. This appears sometimes in textbooks in reading, so it's important to recognize that this could be here. A lot of the time, pro-metaphase is crossed off the list here, and that's what we're going to cover. Pro-metaphase is not something you're going to see in your textbook in meiosis. But this emphasizes another point, is this idea that this process is not some lockstep thing. This is a continuum. It's flowing. If we wanted to create another phase in there, we could. But again, we're usually just looking at these 4 phases within each meiosis 1 and meiosis 2. So let's look at them. Here they are. This is a figure from your textbook. And what we see here this is meiosis 1. What we see is these 4 phases, prophase, metaphase, anaphase, telophase, and then cytokinesis is just lumped in there. And let's look at what happens during each one of these. Now, this is where a lot of the action happens in meiosis 1. So prophase 1, what's happening? Chromosomes are coiling tighter. This makes sense just thinking back to mitosis, right? Well, they have to condense because they're going to be pulled apart. This has to be done in a very organized way. The nuclear envelope is dissolving and dissolves completely at this point. Here's something we did not see in mitosis. Homologs become closely associated. Now, sister chromatids, of course, are paired together, but here homologs also come together. And something happens that's really important here in prophase 1, crossing over, it's called, between non-sister chromatids, so a cross from between homologs. So what is that crossing over? So these homologs will form something often called a tetrad or a bivalent, basically because you have 2 homologs associating. There's these proteins that connect them, line everything up. Remember, the alleles for a specific gene are in the same place in each homolog. So, allele for eye color is here, and there's an eye color allele on this other homolog here. They're in the same place. This structure that connects these is called the synaptoneal complex. So this association is often called synapsis, so the connection between these 2 homologs. Again, this is not something we saw in mitosis. And so here's how this crossing over part of this works. So you've got this tight association of the homologs, very aligned. Little nicks will get introduced into the 2 non-sister chromatids that are adjacent one another. And they're directly across from one another. That's super important, right? That these don't just randomly occur wherever, but they're occurring right across from one another. Here's the synaptoneal complex, these proteins that make this tight association. You've got these nicks here. And then the DNA will actually, this is the crossing over part, the DNA strand from this blue non-sister chromatid will fuse with this red one over here and vice versa. And so this is that crossing. And so you're connecting this one homolog, non-sister chromatid, to this other one here. And so we can look at this in a little more cartoony way here. It's called crossing over. The actual site of crossing over is called a chiasmata. And here we see these two non-sister chromatids crossing over and exchanging a large chunk of DNA here. And the consequence is a recombinant chromosome. Again, all the genes are still there. It's just that the variant that went with this, all the other different genes here, now have the alleles that were on the red homolog and vice versa. So we've mixed things up, shuffled up alleles from different genes. And this is really important for introducing genetic variation in offspring. And as we'll see in later chapters, this also is very useful for genetic mapping, figuring out where things are on chromosomes. So this will come back again. All right, what happens next? Metaphase 1. So we had prophase 1, now we're in metaphase 1. Microtubules attach from the poles here to each homolog. So this is different from mitosis because we saw different sister chromatids having those microtubules attached. But here, the opposite microtubules are attaching to each homolog. And the homologs are aligned at the metaphase plate. And whether or not the red one is over here and the blue one is over here, for any of these pairs is totally random. So this also introduces some variation. So let's see. Let's take an organism here with the haploid number 3, so 3 sets of homologs, so 3 pairs. You could have them orient like this. So all the blue ones are on the left, all the red ones are on the right. Here's vice versa. This really is these two are really no different, right? One cell is going to get all the red ones, one cell is going to get all the blue ones. But here's where we see some variation. Okay, 2 blue over to the left and 1 red. 2 to the right, 2 red to the right, and 1 blue to the right. And you see these different combinations. And because this is random, you can get a lot of different variation. And then the more chromosomes you have, the more different combinations that you can have. All right, so here's what we've seen. In meiosis, we see the separation as we get into anaphase of the homologs, whereas in mitosis, we saw the separation of sister chromatids during metaphase. And that has not happened yet in meiosis. All right, so anaphase 1, homologs separate, sister chromatids still together, and then telophase, the nuclear envelope reforms around the chromosomes. And the other important thing to remember here is those sister chromatids aren't identical anymore because that genetic crossing over has occurred. All right, so where are we at? Telophase and cytokinesis has happened here in meiosis 1. So after telophase 1, the chromosomal makeup of each daughter cell is what? All right, so this one sometimes can be a little bit tricky because you might be thinking, well, our daughter cells at this stage after meiosis 1, they've got those 2 sister chromatids. Therefore, that sounds diploid to me. But you have to think back, what does it mean to be haploid or diploid? It has to do with how many homologs you have. Do you have the pair? Do you have the paternal and maternal homolog? And no, what's happened after meiosis 1 is those homologs have been separated into those daughter cells. Yes, their sister chromatids are there in each one, but you only have that one homolog. And so that's the critical thing here in understanding it. An example I like to give is this shoe analogy, if you're having trouble understanding this. And we'll follow this pair of shoes, right? So you flip-flops. You need to have a left and a right one. Now, think of it like the chromosomes. You got a maternal and paternal homolog. So here's a diploid. Both homologs are present. So during S phase, these things are copied. Yeah, okay, now we've got 2 copies of each. So think sister chromatids. Still diploid, right? Because both homologs are present. In mitosis, those cells divide. Still, each daughter cell has both homologs. So they're diploid. So diploid, diploid, diploid. In meiosis, that first step happens. We copy the DNA. So we've got still a diploid cell. And it's after meiosis 1 where the homologs separate. So this would be the shoe analogy, is that first daughter cell here has got 2 left shoes. So it's, yeah, okay, you look, well, there's 2 shoes there, but they're not 2 different shoes. They're both this left shoe, or here, both this right shoe. And so these, at this point, are haploid cells because they only have 1 homolog. Again, even though there's 2 copies, sister chromatids, they're just haploid at this point. That's what defines that haploid and diploid. Okay, so what happens next? Well, it's meiosis 2, which, as I mentioned before, looks like a mitotic division. Here's the general overview figure from your textbook here: prophase 2, metaphase, anaphase, and telophase, and cytokinesis. And we can look at each step here and some of the details of what's happening. We'll go through quickly. So in prophase 1, that spindle apparatus forms, the microtubules will attach to sister chromatids, and that nuclear envelope will dissolve, and the sister chromatids will start to move toward the metaphase plate, kind of like we see with mitosis. At metaphase 2, there'll be alignment at the metaphase plate, so lined up in the middle here. And remember, this is a point of emphasis, because of that crossing over that occurred back in meiosis 1, prophase 1 specifically, these sister chromatids aren't identical. And you might not be able to see it here, but we see some little color differences on the arms of these chromosomes. Anaphase 2, well, sister chromatids separate. So those cohesins that have held them together get cleaved, and now they can separate and move toward opposite poles. And then finally, telophase and cytokinesis. The nuclei form, meaning the nuclear envelope reforms, the chromosomes decondense, and then we have these 4 haploid daughter cells. And each daughter cell is genetically distinct from each other. Remember, because of that crossing over, it's jumbled up some of those alleles. And because they're haploid and because of that crossing over, they're different from the parent cell. All right, so after telophase 2, what is the chromosomal makeup of each daughter cell? All right, this one should be a little easier and more intuitive. Yeah, they're haploid at this point. So we've been making comparisons of mitosis and meiosis throughout this, but it's worth revisiting this and hitting some of the high points just to drive home these really important differences between the two. So here we have a parent cell that's diploid. This is going to undergo interphase in both mitosis and meiosis. And then in mitosis, prophase, metaphase, anaphase, it's clipped out here, pro-metaphase. That's okay. But we end up with diploid cells. Whereas in meiosis, we're going to end up with haploid cells, and 4 of them. We have this crossing over that occurs in prophase, the random alignment of homologs during metaphase 1. That's really important. And then these 2 rounds of cell division, cytokinesis, I should say, in meiosis rather than just 1. Now, it's another important point is there's not some other DNA replication that's taking place before or after this cytokinesis event. It only happens in interphase. All right, so mitosis is conserving the number of chromosomes, so parent cell identical to daughter cells, whereas meiosis is reducing that chromosome number or halving it from diploid to haploid. And again, meiosis includes those 2 divisions after that original DNA replication in S phase. Driving these points home because they're so important. 3 events that are unique to meiosis that are super important. That's synapsis, so the association of homologs and crossing over, and allows for genetic information exchange. Alignment of those homologs in metaphase is random. It's going to be important, we'll see shortly. And then that homologs are separated in anaphase 1, not sister chromatids. So remembering back to mitosis, during anaphase or at the end of metaphase, we saw the cohesins being cleaved in between sister chromatids. That separated them in metaphase. But again, in meiosis, the cohesins that help connect the homologs are separated in at the end of metaphase 1 and into anaphase 2. So that's what we're seeing here. So here's these homologs being held together. And so they can separate now, homologs separate, and it's not until anaphase 2 that the cohesins that hold the sister chromatids together, we see them coming apart here. Here's this enzyme separase that cleaves those cohesins, then those sister chromatids separate. So why sexual reproduction? You know, if you think of like how a clonal organism reproduces, it can just bud off a new individual. That seems very efficient. So, why invest, in some cases, all sorts of energy into sexual reproduction? So, like, say, attracting a mate, for example, for organisms that do that, or undergoing this meiotic process that's different from mitosis. It all has to do with generating variation, genetic variation quickly. And so where does that come from? Well, ultimately, genetic variation comes from the form of mutations. So little nucleic acid changes, or sometimes could be big. A lot of times they're small individual changes that occur over time. So this is the ultimate source of variation. And so as a consequence, you might end up with different alleles, those variants of a gene. And remember, each of your somatic cells has 2 alleles. And so here's an example here. We've got an allele, this blue allele and this purple allele, whatever they happen to be. Those are different variants. Sometimes you might have the same allele for that particular gene. And so that crossing over and the alignment during metaphase allows for the mixing of these different alleles. So these are important things that contribute to genetic variation. We talked about crossing over. We've talked about this independent assortment of chromosomes. That's at metaphase 1. And then we'll talk about here random fertilization. So let's look at that metaphase assortment here again. And a very simple model. So we've got 2 type 2 chromosomes. The haploid number in this case is 2. And you could have a couple of different possibilities. Again, this is ignoring crossing over. We're just saying that, all right, let's just ignore that. That didn't happen in this case. You could have the chromosomes line up with the red ones to the right, blue ones to the left. And then this first set of daughter cells just has the blue ones, blue homologs. This one has the red homologs. And here's your daughter cells. Another possibility, though, just based on random chance, is the blue one to the left and this red one to the left. And so these daughter cells now, these are genetically different than what happened in scenario 1. And so given the chromosome number, the number of possibilities, ignoring crossing over at this point, we're just talking about that alignment during metaphase, is 2 to the n, where n is the haploid number. So in humans, n equals 23. We have 23 pairs of chromosomes. So it's 2 to the 23. That means there's over 8 million possible combinations just from this alone. Now, of course, we cannot ignore crossing over because this occurs all the time. And this is critical because you're taking alleles that were on, say, together on one homolog, for different genes as we move along the chromosome here, and you're shuffling them with the alleles, again, for that same gene, but that were associated with the alleles of other genes on that other homolog. So it allows this mixing up of alleles, producing this genetic variation. And you can see that here in this example, just with one set of chromosomes as an example. So here's the crossing over. The chiasmata is the site of crossing over. And we end up with these recombinant chromosomes. And you see, but by the color here, we've mixed up pieces and parts of them. All the same genes are still there. They're just alleles are associated with different alleles for other genes. Okay, so which of the following statements correctly describes the characteristics of recombinant chromosomes that we've been talking about? All right, so yeah, again, it has to do with that mixing up of alleles. And if we go back to this figure here, we say have this pair of homologs. Let's say the blue one here was the maternal homolog originally inherited from this person's mother. The red one is the paternal homolog inherited from the father. And what we see here were alleles that were associated with the father, I'll say red here, now are tied with these other alleles that were from the maternal chromosome. And so this allows for that shuffling. On the other end of it here is this idea that, okay, we've produced these gametes. There's all kinds of variation that can lead to those, from the crossing over to 8 million different combinations during metaphase 1 of alignment. But if we think of it another way, when those gametes fuse, you can think, well, statistically, maybe not in reality, but statistically, any gamete could fuse with any other gamete. And the possibilities are staggering. And this is just thinking about gametes from a single individual, from a hypothetical standpoint. So if we had 8.4 million possible chromosome combinations, we're ignoring crossing over here, which adds some variation, producing a zygote with about 70 trillion diploid combinations. So any gamete going with any other gamete, there's 70 trillion different offspring possibilities. So the numbers are staggering, and that's where we see this genetic variation come from. All right, so let's finish up this chapter by putting what we've learned into a broader evolutionary context. And we can think of, why does this genetic variation matter again? Think about it like this. In a stable environment, a world that never changes, clonal reproduction with, some mutations occurring here and there works okay because, nothing's really ever changing. So whatever that creature organism, species has worked out, and it's surviving, it's there, it seems to be fine. But the world and the environment is not static. Things do change. So organisms, groups of organisms, species that have more variation in their abilities or how they look or how they work have some advantages, right? Some of those variants might be more successful in a changed world. Other variants might be less successful. And this is the underlying concept of natural selection. So some are selected for, meaning they better match or more well adapted to a certain environment in that condition. And we're selecting for certain alleles and selecting against other alleles that don't work well. And the more variation there is to work on, the more likely that organism or species, not individual organism, but that species and population is going to sort of get through that changing environment and adapt to that new situation. All right, so keep that in mind as we move on through the textbook into later chapters. And that's it for meiosis. Genetics is next.